Hello, everyone, and welcome to our Q&A session with Dr. Mota. First of all, on the behalf of our committee team, I would like to thank everyone for being here with us today, and we hope you are enjoying our Congress so far. Second, but not least important, I would like to thank Dr. Mota for being here with us, for accepting our invitation on being a lecturer at this year's Congress. I would like to say what an honor it is to be here with you today and to be able to experience a little bit of a chat with someone who has led such an promising and revolutionizing project and research. So thank you so much, Dr. Mota. Before we get started, is there anything that you would like to say?
Well, I would like to thank you and also the organizers, Professor Luciano, for the kind invitation. It's remarkable, the conference. It's really nice and really broad in topics, so that's nice to follow. And thank you for the opportunity to share a bit of the work that we do here in our lab.
It is our pleasure, Dr. Mota. We're very excited. Many questions here. Everyone obviously congratulating your work. But let's get started so we have time to answer as much as we can. The first question is from Nayara. Thank you, Nayara. She says, thank you, Dr. Carlos, for the lecture. How do different bioprinting approaches affect the scalability and cost effectiveness of producing tissue-engineered constructs for widespread clinical use?
Okay, that's a very interesting question. It's still a very puzzling question, just because bioprinting is still truly not massively used in clinics, so the costs are still really high. You have to consider bioprinting as a patient-specific therapy because you need to use almost in all cases patient-specific cells or cells derived from the patient itself to avoid rejection. So it's similar to an organ transplant. So the costs initially, I think they will be high for sure. There is some initiatives, especially here in the Netherlands, where they are trying to do upscaling steps before printing and before creating organs and tissues. And that's definitely something that needs to take into consideration the scalability and the cost, of course. But there is some evident progress on automation strategies to reduce costs. But we have to think that truly bioprinting will always be a patient-specific solution, so it will be intrinsically expensive. On the other hand, 3D printing allows to have off-the-shelf solution, despite that there is always the patient-specific possibility. In some other cases, there might be not needed to be a patient-specific implant. So in some cases, there might be off-the-shelf solutions with 3D printing. But truly, the bioprinting will always be a patient-specific solution. So it will always be, I think, initially quite expensive. So yeah.
Okay, thank you, Dr. Mota, for your answer. Yeah, I'm sure as time goes on, things will get easier. Okay.
Yeah, sure.
Okay, next question is from Juliana. Thank you, Ju. She says, how can the integration of artificial intelligence improve the design and efficiency of bioprinting processes in tissue engineering?
So that's also a very interesting question. Thank you. So we use AI partially, not yet fully. So AI can be used directly during the bioprinting process for process optimization, but it can also be used before and after. So there's also AI tools being integrated, for instance, for trying to predict a bit the outcome of the bioprinting tissue. The bioprinting is a small portion of the full workflow. So there is different AI solutions being explored for different steps of the full workflow. But that's something that has gradually started. It's not truly AI. It is more computational modeling. The true AI requires a lot of data and a lot of information, which gradually will come for sure. But for now, it's still computational modeling.
Okay, thank you, Dr. Mota, for answering the second question. Many questions, and obviously celebrating your presentation. The
Thank you.
next one is from Andre. He says, how does the speaker emphasize the importance of eco-innovation in addressing environmental challenges related to 3D printing technology? And what are some of the proactive measures for preventing future damages?
Okay, so if we are talking about 3D printing, so the use of polymers or metals, I think the true nature of the technology is more eco-friendly because you normally use less raw materials, so and you also create less waste. If you are talking about bioprinting, then the story is slightly different. Despite that there is some new innovation along not really on tissue engineering regenerative medicine, but there is some innovation around food bioprinting, for instance, where researchers are trying to recycle and reuse, for instance, cell culture medium. That might not be a true possibility when it comes to implants for patients. So intrinsically there will be an impact of the technique and the technology itself. Of course, that during our lab and in our research, we try to be as less environmentally impact as we can. But of course, when it takes to when it the process takes into account cells, there's limited things that we can do. We can try to make the best use of the consumables that we have. But in the end, we only or we try to limit a bit the use of materials. But when it comes to the cells, there is definitely some impact when we need to generate millions of cells.
Okay, thank you, Dr. Mota. Yeah,
Yeah.
this is a sensitive topic. There are a few projects in our lab which tries to innovate in 3D printing with recyclable materials, but when it comes to certain projects, that can be a little more challenging. Okay, thank you. Next question from Alessandra. She says, thank you very much for your very interesting lecture. In printing more complex organs that would require a mix of multiple materials, such as softer, harder, and with different functions. Is it possible to mix different printing techniques for a single scaffold? How does your team address this type of complexity?
Okay. Yeah, sorry that the slide where I was explaining the bioprinting didn't show up, so I apologize for that. So indeed, there are several bioprinting principles and techniques that are individualized in one platform. But what we normally have in our labs is platforms that combine multiple techniques. So we have, for instance, a platform that allows us to do continuous extrusion, but also droplet on demand, photo cross-linking, all in the same platform. And that allows us to dispense, for instance, different cells or different bioinks, also have different cross-linking mechanisms throughout the construct, so that can also allow us to modulate a bit the stiffness. So that's the true advantage of bioprinting, is that you can combine different cells and different cell types and different materials on the fly. So you can have really a mimetic tissue being produced. Of course, that is not a mature tissue. So normally what we try to do is to try to emulate a bit the morphology of a tissue that we are aiming for. And then we let the cells to then take the role that they have to do, right? They normally secrete their own matrix, they secrete their own tissue, they reorganize inside of the tissue that we print. So we try to emulate a little bit closer to the tissue, but then the cells play a key role after the printing process.
Great, amazing. Thank you, Dr. Mota. Okay, I think we still have time for one more question. Another question from Nayada. She says, what are the key challenges in creating scaffolds for tissue regeneration that accurately mimic the natural extracellular matrix in terms of porosity, shape, and the material properties?
So there's several challenges, but I think the true challenge is to try to anticipate exactly what happens after implantation in a human, right? So, of course, that there is a lot of preclinical studies that we do before thinking of reaching to the patient. But for instance, the example that I gave in the presentation, where they did 3D printed scaffolds for long bone defect, and this is a clinical study that is happening here in Maastricht. That company has been developing those particular scaffolds, and it's just a polymer, it's a polymer with a ceramic. Those scaffolds took 20 years to develop, to validate, to certify, to do preclinical tests, and then to do first in human. So, there's a lot of challenges, especially on the translation, on the cost still, on having the companies, normally healthcare providers or insurance companies, covering the costs of such surgeries. But there is a true advantage on having these 3D printed or bioprinted implants for sure. But of course, those are the. I think the main limitation is to try to anticipate what will happen after the implantation, because every patient is different. Despite that we try to make it suitable for the patient, every patient will react differently to a specific implant.
Yeah, well, I completely understand the challenges, but I'm sure you can overcome them with time.
Yes.
Dr. Mota, that's all the time that we have for today. We have many other questions that unfortunately we ran out of time, but we will make sure to email them to you. For all the attendees, all of your questions will be sent out to Dr. Mota so he can personally answer them if he would like to. But other than that, Dr. Mota, on the behalf of the committee again, I would like to thank you so much for being here with us today, taking your time on answering our questions, presenting your amazing work. I wish you good luck, and I wish to see you again.
Thank you very much. Thank you for the invitation. It was a pleasure.
Okay, our
Have
pleasure.
a nice day.
You too. Thanks, Dr. Mota. Okay, guys,
Bye-bye.
that is all for our Q&A session with Dr. Mota. Feel free to log into our next presentation starting at 3 o'clock. See you guys soon.